My name is Aamir Mecklai, Managing Director with Bank of America in Healthcare Investment Banking. I had the privilege here to co-moderate the next panel with Dr. Cudkowicz, sitting next to me right here, Chief of Neurology at MGH. So this panel will be focused on genetic medicines and its applications to CNS. While our primary focus will be CNS, we asked the panelists certainly to feel free to talk about other indications as well, just to mix it up a little bit. Rather than doing an introduction for each of us, we'll ask each of the panelists to introduce themselves as part of the first question. And before we jump in, I just want to remind everybody in the audience that feel free to ask any questions through the app and make this more engaging. So to start off with, we're thrilled to have a very diverse set of representatives here from representing various modalities and approaches for genetic medicines. So what I'll ask is each panelist is to start off to introduce themselves and talk a bit about the approach of genetic medicine that you're using and importantly how you're tackling disease.
Great, thank you so much, Aamir and Merit and the organizers of the forum for having me here. My name is Janice Chen. I'm the co-founder and chief technology officer of a biotech company called Mammoth Biosciences. We're based in the San Francisco Bay Area, and the genetic medicine modality that we focus on is CRISPR. So I started my career in the CRISPR field as a graduate student in Jennifer Doudna's lab at UC Berkeley almost 10 years ago. So I've seen sort of the beginnings of the field, starting from a very niche area on bacterial immunity, something that was frankly underappreciated at the time, to now seeing, we're at the cusp of our first approval for CRISPR products. So I've seen sort of the rise of this field and also recognizing both the huge opportunity of the most famous CRISPR enzyme, Cas9, but also its limitations. And so one of the things that we've been focused on over the last decade is really thinking about how do we harness the diversity of CRISPR to go beyond what Cas9 can do. And part of those discoveries through that approach has been uncovering these ultra-small CRISPR-Cas enzymes that frankly are a third the size of the Cas9 system and open up new opportunities for us to deliver these next generation gene editing modalities to different areas beyond the liver, such as CNS and muscle and others. And so we're really excited about this frontier of being able to open up and unlock many unaddressable genetic diseases using this next generation CRISPR technology.
Hi everyone, my name is Olivier Danos. I'm the Chief Scientific Officer at REGENXBIO. REGENXBIO is a gene therapy company. We have a base technology of adeno-associated viral vector. Our proprietary platform allows us to deliver genetic medicines in vivo. We apply this mostly in ophthalmology and in the CNS and also in the skeletal muscle more recently. We've also licensed out this technology to other partners who've developed that to the market today. Notably, there is one genetic medicine that's for SMA, spinal muscular atrophy, called REGENXBIO, which is based on the technology that we own. So this is something that's, we're very proud that this is actually happening and reaching patients today. A lot of progress still needs to be made, but the movement is there for sure.
Hi, I'm Jen Lee. I'm CEO and co-founder of ADARX Pharmaceuticals. At ADARX, a clinical stage company, we develop therapeutics based on RNAi as well as RNA editing. One of our focuses is really developing RNAi-based therapeutics for the unmet medical needs such as Alzheimer and Parkinson diseases. We have exciting in vivo data, but definitely lots of challenges. And I would really share our experience, our journey here with the audience.
Hi, my name is Catherine Stamen-Breen. I'm the CEO of Chroma Medicine. I'm a physician by training. I've worked in biotech for the last 20 years or so across a range of therapeutic areas, including neuroscience. The company that I'm the CEO of, Chroma Medicine, is building epigenetic editors. We leverage the epigenome. The epigenome is the highly conserved endogenous system that regulates gene expression. If you think of the DNA as the hardware, the epigenome is the software. Our epigenetic editors can regulate gene expression without cutting or nicking the DNA, which allows us to avoid a lot of the downstream consequences that can come from using technologies that cut or nick the DNA. The technology is quite flexible. We can activate genes, we can silence genes, and importantly, we can multiplex genes. And so for complex diseases, we can target multiple pathways that lead to disease. The technology is also quite flexible and modular, and so it's scalable and allows us to address a wide range of diseases without having to have a bespoke construct for each disease. We're very excited about the opportunity to address a wide range of diseases, including CNS diseases.
Hi, good afternoon. I'm Nolan Townsend, CEO of a company called Lexeo Therapeutics. We're also an adeno-associated virus gene therapy company. We're focused on larger, rare, and non-rare diseases in the cardiovascular area and the central nervous system area. In the CNS in particular, we're focused on a gene variant associated with Alzheimer's disease, the APOE4 homozygous Alzheimer's disease population. And here we have 3 programs that are in different and complementary ways treating that high-risk population. We're focused really on the genetics of the disease rather than a specific pathogenic mechanism of the disease. We're in a phase 1/2 clinical trial. We've read out low-dose biomarker data showing some very interesting links between the genetics and the biology of the disease, which we can talk more about today. It's great to be here, and thank you for having me.
Thank you for sharing all the exciting work that you are doing and the opportunities. I'd love to have a discussion with the panel about some of the big challenges that are out there and what in particular you and your company are doing to address them. And maybe we'll hear from everyone on the panel, but start with Nolan.
Great. Well, I think I would highlight 2 things. First, the big one in gene therapy is delivery to the central nervous system, getting the right transduction in the brain to the right areas. In particular, for us, for APOE4 Alzheimer's disease, this is a secreted protein, so maybe the challenge is a little bit less significant than it would be for some of the intracellular proteins. But delivery and biodistribution is one of the major challenges, just in general from a technical perspective for the programs we're focused on. Another one I would just say in general in treating Alzheimer's disease is just we have a very large population that we're seeking to treat in totality with Alzheimer's disease, but we see a role for precision medicine within the broader Alzheimer's disease continuum. So we think there will be a continued opportunity to deliver new therapies into this Alzheimer's disease population. However, we do come across the challenge of, are we delivering too much innovation than what society can absorb? And I'm talking about pricing, reimbursement, and access of Alzheimer's treatments. So I think we'd like to find a way through genetic medicines that over time we can reduce the cost of the lifetime treatment for Alzheimer's disease. So I think we will see the introduction of very significant treatments and the change in the standard of care, but we want to find a way that genetic medicines can play a role in ultimately reducing healthcare costs for diseases like Alzheimer's.
Maybe so there are, of course, always a host of challenges in drug development. Maybe I'll build a little bit on something that Nolan just mentioned. A lot of the challenge that can come as you're trying to identify your first indication is, do you go for a very large or a ultra rare disease or something in between? And there's pros and cons of doing both of those. One of the challenges, of course, in going for more common diseases is understanding which the right subpopulation is to address. But the advantage is you have a very large population that you can access as you're running your clinical trials. Of course, the rare diseases, there can be a much larger call to action with advocacy organizations that are creating a very large pull, a large amount of unmet medical need. But for those diseases where you understand the biology, there's good biomarkers, and the population is fairly well described, it can be quite a crowded space. And so it can take actually quite a long time to run your clinical trials because there are often many choices in terms of therapeutics that are trying to address these sorts of diseases. And so trying to choose the disease where you think you can have the most benefit with your technology, but you also have enough access to a patient population where you can validate your technology and prove the benefits can often be a challenge.
So maybe I can follow up on the challenge there. As Nolan pointed out, delivery is a challenge for genetic medicine. And at ADARX, we have developed a platform that can deliver into different deep and uniform delivery to neurons. I think there's an advantage to a simple conjugate versus using AAV or LNP in a way that we alleviate the potential off-target effects from the delivery vehicle itself. So I think that's something that we worked on really, we put lots of focus on in the past couple of years and have developed this unique platform that can deliver directly to deep brains.
So I guess one of the biggest challenge in gene therapy is really the precision with which we can deliver things in cells and which cells we're targeting with a particular vector. In the case of REGENXBIO, we're using adeno-associated virus, AAV, but there are other viral vectors, there are non-viral vectors. What we do is still lacks precision, and this is something that needs to be improved, is being improved. We can do that by, there are several ways of doing that. You can actually act on the vector itself, change the surface of the vector, endow it with properties that it will go to certain cells or others. We know that this is a direction that people are taking and that can be very fruitful. You can actually also act on the way genes are being, the transgene, the therapeutic genes, the payload is being expressed in cells, in certain cells and not others. But again, this is imperfectly mastered. I mean, this is not truly controlled, and we can't, switch off and off really efficiently a transgene, and that brings problems of pharmacology and toxicology, actually. Remember, those are supposed to be one-off therapies, and the modifications, we hope that the modifications we're bringing are going to be permanent. So how do you deal with a toxic dose, for instance, right? You can't just remove it. And so that's a problem, and that's a problem that impacts on also the way clinical trials are being designed, on the kind of discussions that we have with the regulatory agencies. We're doing these kind of things for the first time, most of the time, and that's definitely also a challenge. So that's just to illustrate a few of them. Of course, there are many aspects that are challenging, but again, this is not always drug development as usual. We have to reinvent things just because the concept and the modalities are being different.
Yeah, and my panelists touched on this already, but I think about sort of three big categories of challenges in genetic medicine, at least in the context of CRISPR. The first, of course, is delivery. There's both viral and non-viral delivery approaches, and we all know the limitations in terms of non-viral not being able to easily reach tissues with great specificity beyond the liver, and then with viral approaches, you have the cargo problem in terms of what you can actually fit within that viral vector. So, the way that we think about overcoming that challenge is by starting with a smaller cargo size. You can fit all of that capacity within that limitation. But that still comes with big challenges, and glad to hear my colleagues talk about the efforts and advances in delivery. The second category we think about is also the disease biology. So I think it's pretty clear that right now there's still lots of areas within the genome that are not fully actionable, and I think even from listening to the talks today, be feeling very optimistic about uncovering new disease biology that gives us new targets to actually address when it comes to genetic medicine. I think it's no surprise that even the targets and pipelines that we see today are very crowded. I think a lot of that comes to technology limitations, also is due to regulatory opportunities, but also I think it is a reality of how limited we understand disease of the biology of certain diseases. So I think that's an area that has a lot of untapped potential. And then I would say the third category is just the fact that a lot of genetic medicines, CRISPR included, is a very new technology, and so there's a lot of unknowns here. And I think we are just starting to recognize the power of being able to manipulate areas of the genome beyond just knocking things out, knocking things in, being able to upregulate, downregulate. There's so many exciting ways we can start to modify the genome, and now the question is how do we bring in all that together to make sure that we can address the most critical unmet medical needs.
Thank you. Well, look, I know we spent a good amount of time talking about some of the challenges in the space, so let's try to flip this up to the positive, given there's certainly a lot of opportunity in this space as well, and a lot of excitement in genetic medicine. For example, there could be potentially the first potential CRISPR-approved product between Vertex and CRISPR could be potentially at the end of this year. If you think about some of the opportunities here, and one of the questions that come up all the time is this, one-time gene therapy, how durable these treatments. Many of you guys are working on some interesting technologies to improve upon the durability. So let me start with Catherine in terms of the modality and approach you're using, specifically the advantages you guys have in terms of addressing the question around durability.
So we are developing durable epigenetic editors. We do this by, again, leveraging the epigenome and very specifically leveraging methylation of the DNA. Now, this is the endogenous system that all of our cells use in order to determine the phenotype and function of the cell. It does this for our entire lifespan and through cell division. And so a very highly conserved endogenous mechanism. We think this is a huge advantage in leveraging a system that we know works quite well in all of our cells. I, as a physician, really love the concept of a durable technology. You can imagine for any patient who has a chronic disease, the thought of being able to be potentially cured of that disease for their lifetime is certainly a dream of any physician, but I think for any patient and their families that are confronted every day by a chronic disease, it's really the holy grail for therapeutics. I think particularly for diseases that are diagnosed early in someone's lifetime that they're going to have to carry with them for the rest of their life, being freed of that disease is something that I think can have enormous benefit to patients and to our healthcare system.
Nolan, did you want to add?
Sure.
To that?
Yeah, I mean, I would just expand. I think it's, I think one of the greatest challenges of maybe our generation is the rising healthcare costs. And if you look at therapies today, they are, paid for as they're consumed. And for a lot of diseases, it's for the, patient's entire life. I think with genetic medicines, we have the potential to offer a, single administration, in some cases, functional cure, for the same diseases. So over time, this should, reduce costs for, the healthcare system. So beyond just the therapeutic benefit, I think these modalities offer the opportunity to, fundamentally change the healthcare system and to address this challenge of rising healthcare costs. And I think when we're talking about, diseases that have no existing treatments today, the cost to treat these diseases could be substantial. I mean, think about Alzheimer's and what we're likely embarking upon as a society to treat this population with the introduction of new treatment options. So I think between the types of therapies we're working on at Lexeo and other companies, we believe we have the opportunity to fundamentally change that paradigm with the single administration therapy. And what sits behind that is basically the durable expression of something like gene therapy or gene editing or other modalities that are focused on this.
As a neurologist who's really seen this come to the clinic with our treatments for SMA and Duchenne, adrenoleukodystrophy, and now ALS, it's really transformational. It's so exciting, and durability is key, but the patients are so willing to come in every month as well until it's figured out how to do it with one shot. We touched a little bit about costs, and I was just wondering, maybe Janice, you could start about how are you guys addressing the whole idea about scalability, and how do we develop these in an affordable way for all the possible illnesses that are out there?
Yeah, I think it's a really important question, and yeah, we touched a little bit about the cost of the healthcare system in terms of not being able to provide these curative treatments. So that's one thing that we certainly think about a lot, is having a one and done approach to actually eliminating or actually curing the underlying basis of genetic disease. But the scalability is really important, because if you think about the number of addressable genetic diseases, a lot of those are considered rare disease, and I think there's a big challenge in terms of how can you scale your platform to not just address a small disease burden, but actually can you apply that platform across other diseases, and can you scale parts of your platform to do that? I think the reality is genetic medicines, including CRISPR, are fairly complex, and so I think there are opportunities for public-private partnerships to address the scalability issue. I think a lot of companies try to vertically integrate manufacturing, which I think is a very great strategy, but also I think offers limitations in terms of how can we actually think about putting the best of all of our companies and technologies together to try to address this broader question of not just rare but also common genetic diseases.
So maybe I can touch on the scalability part. I think one approach is, as the panel has talked about, one and done. The other one is that design something very simple, and manufacturing becomes extremely easy, and you optimize that platform technology to have every 6 months or every 12 months dosing. I think that's it's not once and done, but maybe twice a year injection, simple conjugate, deep penetration. I think that's also a great approach, and also non-invasive approach for treating CNS diseases.
Maybe one optimistic comment here on scalability. I think if we look back in the history of medicine, there were other times when we probably felt like a technology or a therapeutic was overwhelming, and when would it ever be scalable? I'm trained as a nephrologist. When dialysis was first introduced, there were very few people that could gain access to that technology. Now, hundreds of thousands of people are dialyzed every year. When antibodies were introduced, there were very few companies that could produce antibodies. They did it in-house. Now antibodies are made on mass quantities, and it's part of primary care now. And so although it can feel perhaps weighty right now from a scaling standpoint, we're just still in the early days of developing genomic medicines. And as we tackle the various components and the complexities of genomic medicines, I'm personally confident that we will be able to scale this so that we will not just be able to treat the very few that have the most severe disease, but we will be able to treat the many with also common diseases that they're carrying throughout their lifespan. And I think we'll learn from the years of developing antibodies, for example, to be able to learn to be able to scale faster and more effectively. I think we have the models in place.
Okay. Good. I think one of the other areas we've seen progress drastically in genetic medicines over the course of the last few years is from a regulatory perspective. The FDA has learned a lot in a lot of these areas of emerging science, and we've certainly seen from the investment banking side of a lot more receptivity and progress made with companies getting through their INDs and getting into clinical trials, and if they get on clinical hold, understanding and how to move those programs back into the clinic. So curious in terms of and I'll direct this question to Olivier, given you've got programs in the clinic. How have you seen the regulatory environment evolve over the course of the last 5 to 7 years, and make it more amenable for the advent of genetic medicine therapies?
Yeah, I think first I want to say that I think there's a lot of goodwill from the agencies that we see, from the FDA, from the EMEA. They do want these therapies to move forward, and they do want to help. Having said that, they can't throw everything out just because this is new and interesting. They have to keep their processes, they have to keep their teams focused on the way they work, and that's also very important. Actually, we wouldn't want this to go away either. So there's kind of a tension here between being very open, reconsidering the way we're doing things, trying to promote more accelerated approval, for instance, these kind of things, and the true process that they have to run every day. It's true that with these new modalities or these new delivery approaches for medicines, we are bringing new problems. We're bringing new situations that they have to consider, and this is most of the time this is done in an interactive way, and this is a discussion, right? So we're not waiting for some message to come from above, and this is something that we discuss, and I think so far it's been pretty productive. Doesn't mean it's easy on an everyday basis. It's a lot of struggle, but by and large, this is a productive process.
So maybe I want to comment on that. siRNA, compared with other genetic medicine method, is a modality that has been commercially and clinical proven modality. So agencies have seen it multiple times. So I would say a lot of our interaction with agency has been favorable, and also we do feel that agency is very data-driven, right? You present enough data, and also scientific rationale, and the response has been favorable. Our lead candidates in clinical now is not CNS, it's targeting a liver disease, and we have been able to move the program rather quickly in the clinic. And also that paves the way for the regulatory pathway for our CNS candidate that's coming up next.
Thank you. We've seen these phenomenal successes in neurodegeneration recently with gene therapies, but we've also seen some failures, and I wanted to get your thoughts on kind of what we've learned about it, particularly Huntington's disease or diseases with repeat expansions like C9 ALS. Are there particular genetic mutations that might be easier than others, and do you take that in account in your company? And then Nolan, if you maybe want to start.
Yeah, I think we're focused in the CNS on secreted proteins rather than intracellular proteins for the delivery challenge, that I mentioned before with respect to AAV gene therapy. So from our point of view, there is a bit of a lower technical hurdle in respect to transducing the right cells and getting the right level of protein expression versus in intracellular proteins. I would say that that's a broad, framework here, but I would also say, in a lot of cases, the technology is advancing very quickly. That can allow us with improved capsids or improved also expression cassettes to begin to tackle some of the more challenging targets. So I think it's just a matter of time for a lot of these when we can start with some of the low-hanging fruit diseases. I put in quotes because it's not, none of this is easy, but we can start there and then progressively move to increasingly more challenging or technically challenging conditions beyond these.
Good. Why don't we touch on disease selection? I know Catherine touched on this earlier, but as you think about disease selection, the big discussion always is do we go after the smaller, rare disease and develop some proof of concept, or do we go after a broader or larger indication, maybe a larger, rare, more disease prevalent? And then you marry that with your technologies, where many companies are developing technologies that allow them to do what others have not been able to do in terms of moving the ball forward. And you think about those two different levers. So curious, as you think about disease selection and what informs you as you embark upon a strategy for disease. Why don't we start off with Janice?
Yeah, I think this is an important question for all companies that have a technology platform, is where do you focus and where do you prioritize? The way that we think about it is where are we most differentiated with our technology? Where can we go that other companies cannot? And in our case, being able to work with these ultra-compact CRISPR-Cas systems really gives us an opportunity to think about not just first-generation CRISPR editing, which is typically knocking something out and then letting the cell repair that and producing an indel, but then sort of next-generation precision editing, which involves really carefully targeted changes to the genome. So this can include rewriting, changing specific bases, and so on. So one benefit of these ultra-compact systems is that you can, again, package all of that into a single AAV, making the complexity of the problem much lower, right? You can now take all of that and deliver that to targeted tissues and organs of interest. So with that perspective, we think kind of at the end game, right? Here's where we can go and address diseases that other companies cannot because of the complexity of delivery. And then we sort of say, okay, where are we today in terms of maturity, and what are the logical steps that we must take from a both pipeline perspective, not just the disease, but also the pipeline in terms of our portfolio of modalities that we have the benefit of working with. So we have this sort of multidimensional problem that we think through, and it's about, making sure that along that sort of progression of the technology maturing, but also addressing the diseases that we can uniquely work on, and how do we build that roadmap to getting there. So that's sort of broadly how we think about the different dimensions.
Maybe I'll talk on the disease a little bit. So one of our lead candidates to target tau protein for Alzheimer's disease, definitely this is a large relatively, for CNS large disease indication. And I think we are using our technology to its advantage. For example, if we use MAPT to target, or antibody approach to target tau protein, it can only control the extracellular tau protein. Our way is to silence the messenger RNA so that we, in the end, we reduce or greatly reduce both the intracellular tau tangles as well as extracellular tau protein. And that really, I would say, bring the hope of disease modifying for Alzheimer's disease. And we have seen really great data at preclinical models at this time.
Well, let's talk, we talked about disease categories, but maybe about patient age a little bit. And is there some data that maybe younger children react differently to older adults, to treatment modalities, as well as obviously the question of long-term treatment? And does that factor into your decisions about which disorders you go after? And maybe Olivier, you want to start with that?
No, especially in severe neurodegeneration, as you know better than I, once you reach a certain point, there's basically, it's very difficult to imagine how you would go back, right? And this young patient start for the first year or two to be rather normal, and then they develop normally, and then they fall down a cliff. If you intervene before they fall down this cliff, you can definitely see pretty striking evidence of efficacy. If you're beyond this point, then it starts to be more difficult to interpret. It starts to be, and so the clinical endpoints become very, very complicated to look at. So targeting the right patient at the right age is absolutely key, right? It's absolutely key. And whether you can do something at a later age is going to be very, very disease specific, and even within the disease, I guess patient specific, because it's all about, each patient now is going to evolve in its own particular way. When you're in the context of a very rare disease, the problem is that it's very difficult to have a true natural history study that tells you that has a lot of predictability for individual patients. So you're getting into difficult problems. So to really get the clinical proof of concept that your therapy is going to work, in I think all the examples I can think of, they're being obtained in much younger patients, at least in patients that are pre-symptomatic, not, and depending on the disease, that can be more or less young, but the pre-symptomatic patients are the patients where you want to ask these questions.
Good. I want to go back to a topic we touched on earlier, is around delivery. There's certainly been a lot of progress in the genetic medicine field, but probably others would say the field of delivery has not progressed as far. So curious, just in terms of what you're seeing in the areas you're focused on, where some of the advances are in delivery and how some of those advances in delivery can be used to accelerate your strategies in the disease space you're working on. Noah, why don't we start with you?
Yeah. So I think historically, and let's first talk about CNS. Typically for gene therapy, you would utilize a central nervous system route of administration, like intrathecal, intracisternal, or intraparenchymal routes, and the capsids were designed, the expression systems were designed around those routes of administration. Increasingly, there are capsids that are emerging that are IV administered but can transduce the CNS, which is a major change versus what existed in gene therapy previously. And so I think this is what's evolving on the CNS side of things. And I think over time, over the next 5 to 10 years, we'll probably transition from, in most cases, CNS routes of administration to more IV administered CNS gene therapies. There's also some other changes occurring for other organ targets. We're focused on cardiovascular disease. There's a lot of development in the recent years on transducing the heart, which has been a notoriously challenging organ to, let's say, deliver genetic payloads to, and the technology there is advancing pretty rapidly as well. So I think across several different organs, you're beginning to see these rapid advances in delivery technology.
Can I make a quick comment just on the notion that delivery technology hasn't really evolved? I think it has hugely evolved. I mean, even within the past 10 years, it's unbelievable what we can do now and we couldn't do before, right? And if you now look at 25, 30 years ago, we just couldn't do anything in a mouse. I mean, that was at this level. So it may not be fast enough for certain things but it is progressing a lot. So and it's still progressing every day. We see that at every each conference brings new things. It's never fast enough, but it does happen.
It's always never fast enough.
Talking about not fast enough, there is a question from the audience about the studies the panelists are doing in dementia. If you could maybe specify a little more the types of dementia where treatments are in development and when these treatments might become more widely acceptable in the society. And what do you think is the most promising in dementia? We're all worried about this. So. Nolan, you raised your hand.
Sure. Well, we have an ongoing phase 1/2 study for APOE4 homozygous Alzheimer's disease patients. We've read out low-dose biomarker data showing a reduction in several of the biomarkers commonly associated with Alzheimer's disease versus baseline, so amyloid beta, tau, and phospho-tau. So it's early data. It's our lowest dose, but we thought very interesting. So this is the therapy that we have in the clinic. We'll complete enrollment of the study this year. We'll have a data readout of the entire study next year, and hopefully this will demonstrate proof of concept and have us moving this to a later stage clinical study. So this isn't a distant therapy, a gene therapy for Alzheimer's disease. This is in the clinic now. Patients have access to this clinical trial, and hopefully we will see over the coming years a gene therapy such as this that can treat this subpopulation of Alzheimer's disease patients.
Anyone else? Zen?
Yeah, so we target MAPT to reduce tau tangle for Alzheimer's disease. Right now, in preclinical studies, we have seen exciting data. Next year, we probably, if things goes really on schedule, we should be starting clinical studies on that. So there are lots of technologies, obviously companies, biotechs that are tackling this area, and I'm really optimistic that some cure or some treatment will come up in the next few years.
Okay, I know we're running short of time, so just one last question. I'll direct this to Catherine. Just what do you see in the next couple of years that you're most excited about?
What do I see in the next couple years that I'm most excited about? I think it's a little bit of a combination of things. Maybe what I'm most excited about is maybe a little bit more of a general statement. I'm really excited about how many different technologies there are that are taking shots on goal against a broad range of CNS diseases and diseases outside of the CNS, but particularly for this forum. CNS it's a tough area. It's been difficult to deliver. The biology is complex, and the broad opportunities right now in genomic medicine. It's just an incredibly exciting time to be in medicine and in drug development. I think on some level we all root for each other, maybe on every level we root for each other, because a win in one of our technologies in a particular disease is a win for all of us. And so I'm just excited to see the continued cards flip so that we can learn a little bit more about the biology of the diseases through genomic medicines, and we can develop therapeutics that are going to fundamentally change the lives of patients that had very little hope. So that's what I'm most excited about.
Thank you. Well, I want to thank this panel and end with actually a quote from the audience about how wonderful this panel is and that we should go on a tour around the world and share your knowledge. So thank you for sharing it with us today.